We are back for our third and final installment of the immune system. I guess the fourth, technically, if we remember that we had that conversation about COVID with Laureen Campana and Misha Miller, which if you haven't watched yet, definitely do. It's full of really great stuff. So, have some breaths. We're gonna move forward with cellular immunity as soon as I share my screen. Okay. Immune system and COVID-19, where are we at now? We have worked through your innate immune system, talked about inflammation, inflammatory process, how that helps your immune system fight off infections and get ready for healing. And we also, last time we were together, talked about humoral immunity, which has to do with the release of those antibodies into your humors, which would be the blood and lymph system, and how those help your body to fight off infections. And we started talking about how your immune system gains memory and how vaccinations work. So on our docket today, we are going to be talking about cellular immunity as part of your adaptive immune system. And specifically, we're going to focus on the cytotoxic T cells. As you'll see, there are multiple types of T cells, but we're going to do most of our learning and work with those cytotoxic T cells because they are particularly important for fighting off viruses such as COVID-19. And we will complete our discussion talking about allergies and the autoimmune system. A lot of you had questions about allergies and autoimmune system. Spoiler alert, a lot still not known about allergies and autoimmune system. So if that's an area you're interested in going into, you will have loads of research and questions to ponder because there's still a lot unknown about those. And we'll close with how to help your immune system, which will include a piece about sleep and your immune system, what sleep deprivation specifically does to your ability to fight off viruses, to try and help inspire you during this pandemic to get plenty of sleep. We've talked about innate defenses and adaptive defenses. Last time we were together, we were talking about that humoral immunity, which is your B cells, which are producing antibodies for different cells in your body to recognize those flagged cells to help kill them, as well as neutralizing viruses. Remember that chart? And then today, we're talking about cellular immunity and how that works. And the big players in that are your T cells. So whereas your innate immunity, we were talking about how there are just some phagocytes. Do you remember which ones those are? Your macrophages and the neutrophils, yep. And then the other one was your natural killer cells, and they don't do phagocytosis, the natural killer cells, but they all work to just generally kill bacteria, viruses that seem to be causing problems, are kind of targeted as just a general nuisance. But now we're getting into cellular immunity, meaning that these T cells, specifically the cytotoxic T cells, cyto as in cell, toxic as in poisonous, deadly. So these type of T cells are actually using this conversation with antigens to specifically target unhealthy cells of the body and to kill them. And one of the things that they're going to use in order to target those cells are those MHC1 proteins. We talked about those last time we were together. So where are you at with your understanding of MHC1 and 2 proteins? So remember, they look kind of like this, those U-shaped proteins that hold the Cheeto-shaped antigen in them. And do you remember this? So you have two types, 1 and 2. So which one is true about MHC1 proteins and how they display antigens? Do they A, engulf their antigens through phagocytosis and then display them? Or do MHC1 proteins B, display antigens that are made within the cell? If you said A, you are wrong. MHC1 proteins are the ones that are going to display the antigens that are made within the cell, and those antigens can be self-antigens that present to the immune system that, hey, we are part of this body, please pass us over, do not kill us, versus versus if that cell is infected with a virus and that virus is making proteins within the body, or the virus is taken over the mechanisms of the body to make proteins, which would be more precise to say. But the proteins that are just being displayed by the MHC1 are viral proteins, and then that becomes a flag to the immune system, specifically these T cells, the cytotoxic T cells, that this is a cell that's been invaded, it's unhealthy, it needs to be killed in order for the rest of the organism to be healthy. Versus those MHC2 molecules, and those are the ones that do A right here. They're the ones that are displaying antigens through phagocytosis. So things like the dendritic cells and macrophages. Oh, there you go, there's our answer, B. So now that we've reviewed that, let's look at this figure from your text. And you might perhaps instantly recognize this is a little more complicated than the flowchart we looked at for the B cells. These are the T cells. So the T cells actually got their name because they mature in the thymus, which is a endocrine gland that sits basically on top of the upper portion of your heart. And these lymphocytes would still be procured or generated in the red bone marrow. So that cellular division is happening in the red bone marrow. And then those immature lymphocytes are going to leave the red blood marrow through the cardiovascular system and make their way to the thymus. And the thymus is where they grow up. And they become one of two things. They either become what's known as a CD4 cell, or they become a CD8 cell. And the difference between CD4 and CD8 at this point is the type of receptor that they've got on their plasma membranes, which is shown in slightly different shapes between CD4 and CD8. So let's follow CD4 for a moment. CD4s become activated when this receptor interacts with some sort of antigen. And in this case, you'll notice on the CD4 side of the house, we're talking about those class 2 MHC proteins. So these are antigens that have been incorporated into the APC. Remember that one? That's the antigen presenting cell. So one of the antigen presenting cell types is a dendritic cell, and these types of cells are really important in just training your immune system. The dendritic cells are moving throughout the body, just monitoring things in general, and then they're going to pick up different antigens with that great surface area to volume ratio. As they migrate to the lymph system and hang out in the lymph nodes, now they're going to be displaying different antigens to different T cells that can get activated by that interaction between the T cell receptor and the antigen being displayed by the class 2 MHC protein. So when that interaction happens, then a few things can happen to this cell. One is it can become a memory cell. So similar to our B cells, remember those, your B cells either matured into plasma cells, which were the cells that were making antibodies and releasing them into your humor, your blood, and your lymph, or they became long, and those cells live 4 to 5 days, and then they go through apoptosis and die, or they become a really long-lived memory cell, and those memory cells can live the rest of your life circulating through your body, carrying the memory of that former infection that helps ramp up your immune system really quickly if you get infected with that same bacteria or virus that's carrying the same antigens again. Same thing for your T cells. You can have CD4 memory cells that are going to be long-lived memory cells that help boost your immune system if you're ever infected with this particular antigen again. If they don't become memory cells, then they become different types of effector cells. Effector cells meaning that they are affecting your immune system. They're causing an outcome. And those either become helper T cells or regulatory cells. And we'll talk a little bit about these guys. For a brief overview, what are they doing? Well, the helper T cells help other cells basically become activated and help the immune response to occur. Your regulatory T cells play a different kind of role in that, as you've learn from Laureen about cytokine storms and how we'll be talking about in autoimmune system diseases, the regulatory T cells are trying to help your immune system not do that kind of stuff, to not damage the self cells. And so they're helping to regulate things amongst other jobs, but that's one of their primary jobs. But we're going to spend most of our time over here talking about these CD8 cells. So it's in this immature state that we use the term CD4 and CD8. So CD4 are going to mature into either memory cells or helper cells or regulatory cells, whereas a CD8 cell, that's going to mature into our cytotoxic T cells. And let's look at how they get activated. So you'll notice here that this is a different class of MHC protein. They're getting activated by class 1. What does that mean? Well, as we just talked about, class 1 antigens are the ones that are being generated inside of a cell. So the job of these guys, they've actually already gone through a training previous to this. If they make it to this spot in their training as a cytotoxic T cell, they go through a training where they are shown self-antigens, and the cells that react to those self-antigens are killed. They go through apoptosis, they're dead. So that's one of the ways your body's trying to avoid autoimmune diseases, is if you do make a T cell that has a receptor that reacts with a self-antigen, if all goes well, that thing dies and it never reacts with your own cells. So those are the cells that get to this point of activation inside of the lymph system. And so we've gone from the thymus into the lymph system, and now this maturing cell is going to get activated by interacting with most often a viral antigen that's on this MHC, but it can be something else. It can be a protein bit from a cancerous cell. So cancer cells make different proteins than healthy cells. So it could be one of those, or it could be something else. But for our intents and purposes, since we're talking about and focusing about COVID-19, let's say that this is a viral antigen. When our cell interacts with that viral antigen, it's going to activate it. It has one of two fates. It either becomes a memory cell that's going to help with that second immune response, or it's going to divide and become a cytotoxic T cells. So we're going to talk about how does that happen, or rather, how does a cytotoxic T cell end up killing a cell that's been infected with a virus? So we talked about this friend, your dendritic cell, and its incredible surface area to volume ratio, where it can display all kinds of antigens. That is going to they're going to be displayed, right? So something like a macrophage can display things with MHC2s. Your dendritic cells are pretty awesome in that they can use both MHC2 and MHC1 proteins to display the antigens that they've encountered. So let's watch an animation about how your cells are doing their jobs. Come on, YouTube, you can do it.
The adaptive immune system provides a more targeted defense.
Oh, wait.
Dendritic cells are like intelligence specialists.
Whoa, stop. Oh, am I still recording? Oh, nice, Niggly. What fun would be a video without any technical difficulties? My apologies.
The adaptive immune system provides a more targeted defense. Dendritic cells are like intelligence specialists. They collect pieces of the invader at the site of the battle. Then they travel to the nearest lymph node, triggering a cascade of events. Inside the lymph node, the dendritic cell presents pieces of the invader to T cells, the immune system's communication specialist. In turn, T cells activate B cells, training them to form antibodies, the molecular weapons that target the invader specifically. The immune system now mounts its ultimate defense. B cells release millions of antibodies, each antibody's shape tailored to identify and neutralize the enemy. Right. The immune system deploys these tailored weapons.
So I wanted you to notice this time that when we get to this point here, and this dendritic cell is hanging out with this T cell, did you notice what kind of T cell it was? Do that one more time. Oop.
In turn,
Oop.
Dendritic cell presents pieces of the invader to T cells, the immune system's communication specialist.
Oh, a helper T cell. So is that a CD4 or a CD8? Yeah, the CD4s, right? So these are talking to, so antibodies are being made, can be made through the MHC2 complex. I think they can also be made through the MHC1 complex. Okay. Back to it. I realized we haven't reviewed the lymph system, so maybe we should take a moment and remember what is this lymph system and what is it doing and how does it talk to the rest of the body. In the systemic circuit, so the side that's feeding your body oxygen and nutrients, we have arteries become small arterioles, then we have our capillary beds, and then blood's going to return through the vein. Integrated, interlaced with those capillary beds are these lymph capillaries. And you'll notice they kind of end, right, like a cul-de-sac. They're not making a loop in your body. But you have these lymph capillaries that are able to, based on differences in pressure and osmotic gradients, basically they are picking up the interstitial fluid that has leaked out. So the blood plasma that leaks out of the capillary beds, now we call it interstitial fluid. Some of that actually goes back into the capillary bed, but a portion of it does not. A portion of it stays as interstitial fluid. And the way that gets picked up so that there's not edema happening all over the body is it gets picked up by this lymph system and gets carried back into the blood system. So, but once it's in these lymph capillaries, then those lymph capillaries lead to these lymph nodes. And we have lymph nodes all over our body, but there's certain places where we have more lymph nodes than others. We have several in our neck, which if you've ever been to the doctor, or if you've done this as a practitioner where you feel someone's neck and what they're looking for is, are those lymph nodes swollen? Because when somebody's fighting an infection, that becomes the traffic information center. All these cells are being shunted there to try and fight off this infection, and so they become swollen. And that's one way you can check to see if somebody's fighting off an infection is, are those lymph nodes swollen? Eventually, all of that fluid, once it's gone through the lymph nodes and been checked, that's also right where that's where your T cells are being activated by these dendritic cells or these antigen presenting cells. Once that fluid's been sorted through, picked through, it does drain back into the cardiovascular system. So there are ports from the lymph system back into your cardiovascular system in your neck, basically at the base of those jugular veins. So now we reminded ourselves about MHC1 and antigen presenting cells and how they're working. Let's talk about how cytotoxic T cells become activated and target infected cells. So we'll review this image after we've watched this animation, which is going to go much more smoothly this time. Famous last words, Niggly. All right. So I chose this animation because I think it does a great job of giving you an understanding what's happening at the cellular and molecular levels. Molecular meaning what's going on in the membranes between the different cells, but it has some funky background noises. We'll see if you love it or if it skeezes you out. All right.
T cells. Virus-infected cells are killed by cytotoxic T cells.
Yum, 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 yum.
Cytotoxic T cells contain toxic granules. Their contents are transferred into a sick cell, causing death. The majority of T cells circulate in the blood and lymph. Most are not cytotoxic and are considered naive. High concentrations of naive T cells can be found in lymph nodes, which are scattered throughout the body. Shown here are naive CD8 positive T cells inside a lymph node. These T cells have the potential to become cytotoxic when activated. The body creates millions of variant T cells, each with the unique ability to recognize different pathogens. But T cells cannot recognize pathogens on their own. They first encounter the pathogen via another immune cell called an antigen-presenting cell. Antigen-presenting cells are one of the first cells to detect infections in our bodies. Their role is surveillance, inspecting their environment and taking samples back to nearby lymph nodes. Once in the lymph node, the antigen-presenting cells search for a T cell that will recognize their cargo. By chance, this T cell recognizes the cargo carried by this antigen-presenting cell. Recognition is controlled by molecular interactions between cells. The antigen-presenting cell produces proteins called MHC on its cell surface. The MHC contains a virus fragment. The virus fragment is also known as a virus peptide. Each naive T cell produces a unique receptor that can recognize different virus peptide MHC combinations. The presence of the CD8 molecule defines this subclass of T cells. The receptors on this particular T cell bind to the virus peptide MHC complex. Co-receptor CD8 binds the complex. Receptors cluster. Eventually leading to T cell activation. The activated T cell divides and produces clones of itself. The cells gain cytotoxic capabilities as they grow. Chemical cues attract the cytotoxic T cells to the infection. The cytotoxic T cells specifically recognize the infected cells. The recognition is controlled by molecular interactions between cells. The infected cell displays the same virus peptide MHC complex. T cell receptor binding and clustering trigger the killing process. Cytotoxic granules are transferred and killed the infected cell. Only infected cells display the virus fragment on their cell surface, ensuring that only infected cells are killed.
Be fun. So, as a review, here's our antigen presenting cell. So that could be a macrophage, that could be more often a dendritic cell that we've been talking about. And with its MHC1 complex, it's going to be presenting this antigen to our CD8 immature, naive. You're so naive, you haven't been exposed to anything. So now it does get exposed to something and it becomes mature. Gets exposed to the antigen. There's, of course, some other complex things that are happening that aids this, this activation to happen. Once it becomes activated, it can become a mature, it's going to start dividing, and those are going to become mature cytotoxic T cells. They're full of these vesicles, these granules that are full of molecules that can cause cell death. So, role of helper T cells in all of this. Check it out. This one's 21.18b from your book. You'll recognize this guy. Here's our dendritic cell. And it's interacting with, here's CD8, That's going to become our cytotoxic T cell. But there's something else going on here too. Here's our helper T cell. So remember, this was our on the CD4 side of the house. And it can recognize these class 2 MHC proteins in our dendritic cell. And when it's bound to this dendritic cell, it's going to, this arrow, right, it's going to start releasing what are called co-stimulates dendritic cell. So it's going to stimulate the dendritic cell to express co-stimulatory molecules. And that's what these guys are. So it's going to basically help this to bind more tightly. And this is also releasing IL-2. IL-2 is one of those cytokines that helps with the inflammatory process. What's the big idea here? Well, dendritic cell can now activate CD8 cell with the help of interleukin 2, which is a cytokine secreted by the helper T cell. So when we talk about how there are helper T cells, in this case, in order for a CD8 cell to become activated to mature into a cytotoxic T cell, there has to be a helper T cell around to help that happen. So it helps our CD8 naive cell to mature into its cytotoxic awesome self. Okay, so let's walk through this one together. I'm just going to read it through and then I'll add stuff when we're done, but let's get through there. So here is our mature cytotoxic T cell, and it has left the lymph node and it has found the site of infection. And in step 1, our cytotoxic T cell, that's what TC stands for, identifies foreign antigens on MHC1 proteins and binds tightly to the target cell. So in this case, our target cell, what would that be for COVID-19 in your lungs? That is right. That would be your type 2 alveolar cells. Type 1 are just all the cells in your alveoli that are doing the oxygen carbon dioxide exchange. Type 2, you'll remember their job is to make the surfactant for your alveoli. And the virus, to get into the cell, is recognizing which receptor? The ACE2 or the ACE2, depending on which animation you're watching or who you're talking to. But same thing, potato, potato. ACE2 receptors are how the virus gets in. Once it's inside of the target cell, then it's going to start, it's taking over the ribosomes of the cell. That cell is going to start making viral proteins. Those are going to be picked up by the MHC1 proteins on the target cell, and they are going to be displayed on that target cell. And that MHC1 with the viral antigen is, of course, then what our T cell is going to recognize. So once it does that, then we're going to fast forward to step 2, where the cytotoxin releases perforin and granzyme molecules from its granules by exocytosis. So this granules, that vesicle, exocytosis, we've talked about that one. It's releasing these, and what does perforin do? Well, perforin are a series of proteins that make a little pore in the membrane of the cell. And that pore is large enough that basically anything can go in and out of the cell, and that can cause the cell to die because it can lose a bunch of stuff, or a bunch of stuff can come in and it explodes, right? So it's either going to implode or explode, but it's no good for the cell. So 3, perforin molecules insert into the target cell membrane, polymerize meaning that they are a bunch of small pieces that come together into one big tube, but it's a microscopic tube, but it's on the scale of things, it's large and that lots of things can go in and out. And they're going to form transmembrane pores. So transmembrane meaning that they go across the entire membrane of our target cell, or cylindrical holes, similar to those produced by complement activation. So complement's one of the many complex things we haven't gone into big depth in our lectures. You can read about it more in the book, but complement is a protein that leads to the same action, and that we're just poking holes in the membrane, and if the holes are big enough, things leak out, things come in, cell dies. Number 4, granzyme enter the target via the pores. Once inside, granzymes activate enzymes that trigger apoptosis, which means cell death. And 5, our cytotoxic T cell detaches and searches for another prey. Dun, dun, dun. So they're kind of like zombies. They're clones of each other. It's kind of like eating brains. Anyway, pretty cool. So let's walk through table 21.6 from your book. It's a nice summary of different immune cells in your immune system. So we've got 2 big columns here, the class 1 and class 2 MHC proteins. So remember that those class 1 MHC proteins are displayed by all the cells in your body, or all the cells with a nucleus inside of your body. So things without a nucleus would be, for example, your red blood cells. And they're all going to have, if it has got a nucleus, it's presenting MHC proteins. Those proteins are bound to an antigen. If those antigens are self cells, all is good, you'll be passed over. If those antigens are from, let's say, a virus, then that targets you for cell death if a cytotoxic T cell has been trained for that specific antigen. So if it's been trained for that specific antigen, then it can attack those self cells. So naive CD8 cells, right, they become mature by initially recognizing that antigen on a antigen presenting cell. So in the case of a naive CD8 cell, those are going to be inside of your lymph nodes. And this is the conversation that they're going to have. They're going to be talking to a antigen presenting cell, like a dendritic cell. You don't want to kill the dendritic cell because that's like killing the messenger. The dendritic cell is just there to tell the immune system who the problem is. The dendritic cell itself is not the problem. So if the cell is an APC, meaning a antigen presenting cell, or if it's the messenger or the dendritic cell, here's the message. I belong to self, right? I'm a dendritic cell, but I have captured a foreign invader. This is what it looks like. Bleh. Kill any cell that displays it. And so this is the conversation that a naive CD8 cell would have. Like, let's train this guy up. You could also have that conversation could also happen with cytotoxic T cells as kind of like a memory enhancement. But most of the time, that's what's going on. Then the cytotoxic so this CD8 is going to mature into a cytotoxic T cell. That cytotoxic T cell is then going to leave the lymph nodes, start wandering through the body, and it's then going to have this conversation, right? If the cell is not an antigen presenting cell, meaning it's not a dendritic cell or a macrophage that's trying to serve as a messenger of training your T cells, then it says, I belong to self, but I have been invaded. I shall be sacrificed. Come, or I've become cancerous. Please kill me. Put me out of my misery so I don't do damage to the rest of the body. And of course, it's MHC class 1. So we're talking about endogenous intracellular pathogens. So things that have invaded the cell, like a virus, or if the cells become cancerous. Cytotoxic T cells can also take care of those. Class 2 MHC proteins. So Also, we've got, APCs, antigen-presenting cells like dendritic cells, macrophages, B cells. Those are going to be using those class 2 MHC cells. Dendritic cells can also use MHC1s. But these guys are picking up bacteria and viruses that are just moving around inside of the blood plasma or the lymph or interstitial fluid in between cells. And they're going to pick up those antigens from the exogenous, right, or extracellular pathogens. Who recognizes those? Well, here's where our, CD4 cells, which will mature into helper T cells. Those are the ones that are talking to those MHC2 class proteins. Remember, we need these helper T cells in order to train up our cytotoxic T cells as well. And the conversation they're having with this dendritic cell or the antigen presenting cell is, I belong to self, I'm a messenger, but I've captured a foreign invader. This is what it looks like. Help me launch a defense against it. And those helper T cells are then going to help train up those B cells as well so that they can make antibodies. Cytokine storms. Okay, so clunky transition to cytokine storms. If you haven't already watched the cytokine conversation lecture with Laureen Campana, where she very expertly explains cytokine storms, I encourage you to do that now or when you're done with this. But here's one of the issues that happens with COVID-19 that is being seen. Your immune system gets started. It is picking up this virus. Those dendritic cells are training up your cytotoxic T cells, amongst other white blood cells that are getting involved in this response. And they're starting to launch an attack on the cells that have been invaded. As those cells start to do that, they're going to release cytokines. And cytokines here are in green. And part of the job of those cytokines is to bring in more white blood cells. So that, because there's infection, there's some problem, we need some more help. So those cytokines pull in and help activate more white blood cells. More white blood cells arrive on the scene. As more white blood cells arrive on the scene, they're going to release more cytokines. More cytokines are going to pull in more white blood cells. There's a nice key down here if you want to check it out. So it becomes this positive feedback loop where more white blood cells release more cytokines means more white blood cells. And it just goes around and around in this loop until you get into the state of a cytokine storm where the white blood cells, your immune system starts to do damage to self cells. And that this inflammatory response has become so massive that in the case of COVID-19, it's taken over not just a part of the lungs, but the entire lungs become filled with fluid. It's a component of this, the acute respiratory distress syndrome, where the alveoli become filled up with fluid. Oh, can you diffuse well through all of that barrier? No, not so well. And you also start to get scar tissue on the alveoli. And so now they don't expand and you can't inhale and exhale as well. So they're losing their elasticity. And as you build up the thickness of the alveoli, gas exchange doesn't happen as well either. So what the body then needs to do is to get the infection taken. Oh, let me try this on the couch. I'm gonna fall off the couch. Lisa's using the office. Okay, with the spinny chair. No, I've totally forgotten. Stick with me. Wait for it. Right. So, so the cytokine storm can lead to this acute respiratory distress syndrome. If it's bad enough, then you get put on a ventilator, and the hope is that you're getting enough oxygen during this time where your body's starting to recover from the cytokine storm and the acute respiratory distress syndrome. So the body has to move out of this hyper immune response, reduce the inflammation, pull out all of these fluids, and start to repair the cells that have been damaged. It takes a while, as you can imagine. Hooray, cytokines! So lots of different cytokines that have lots of different jobs within the immune system. But just knowing that you have these family of molecules, their job is to promote the immune system. You also have some cytokines, and their job is to okay, party's over, let's wrap this thing up, we're done. And of course, if those are out of balance, things aren't going so well. And Laureen talks about that nicely, but just a recognition that if it hasn't occurred to you yet already, on a molecular scale, your immune system is marvelously complicated and pretty fantastic. Laureen also talks about some specific foods that help help your immune system and have some specific connections with some of these cytokines. All right. All of that to say, with all of this going on, one of the reasons why our immune system goes wrong is because this is NASA. This is their ground control. You have no central control for your immune system. It is cells talking to each other through molecules like cytokines and antigens and MHC protein receptors, but there's no brains to our immune system. And because there's no control center and you just have cells interacting with each other with these protein conversations, then they're just reacting to this immediate conversation that they're having and doing what they're being told to do within this tiny little molecular conversation. And so you can see how things can go wrong when you don't have a mission control center. What's handy about not having a mission control center for your immune system is that you can have very specific local responses and take care of infections. So when it works well, it's awesome. And most of the time it does. But when it doesn't, then it can get carried away into cytokine storms and some other issues. So let's talk about those. One of those is called low-grade chronic inflammation. And I wanted to talk about this because more and more it seems that the major diseases that cause issues in Western society and growing throughout the world are, the root of them seems to be this chronic inflammation. So what is chronic inflammation? Well, acute inflammation is what we've been talking about. You have some sort of infection or you have some kind of injury that requires your body to heal itself. And so it has this inflammatory immune response. If that doesn't completely resolve and you still have this trickling, let's say, of cytokines moving throughout your entire body, now that can lead into this chronic inflammation that can be throughout the entire body. And your body might currently at this moment be in a state of low-grade chronic inflammation and you really wouldn't necessarily know it. But the issue with that is that it can contribute to things like cardiovascular disease and neurological diseases. So things like Alzheimer's, they think the base of that is long-term chronic inflammation. Autoimmune diseases, things like multiple sclerosis, rheumatoid arthritis, cancer, right? Another one of those diseases that they think one of the bases of it, one of the contributing factors to it, is chronic inflammation. So how do you get your body to not be in this state of chronic inflammation? You need a healthy immune system. We'll be talking about that momentarily, but you can imagine what those things are. It's what we've been told to take care of our bodies on a regular basis anyway, right? You exercise regularly, you make sure you get enough sleep, hopefully not during this lecture, but whatever. If you need it, you need it. You can come back to it. It's being recorded. So you're getting exercise, you're getting sleep, and your diet is full of vegetables and fruits, which are full of all the nutrients your body needs in order for your immune system to be healthy. And you also manage your stress well, that you have managed to have a healthy relationship with stress. It can motivate you to get some stuff done, but you're not carrying it around with you all the time like luggage, and you've got some great social interactions in your life that help you manage that. Okay, so your immune system can go wrong in a few ways, right? You can have these big cytokine storms that blow up. You can have chronic, which is an acute thing. You can have chronic inflammation, which can lead to long-term chronic diseases. There are also autoimmune diseases. So autoimmune diseases are particular diseases where your own immune cells are attacking healthy self cells. So somehow that conversation that's happening, like for example, between your cytotoxic T cells and your self cells, something goes wrong. So now they're starting to attack your healthy cells instead of just cells that have been infected by a virus or just cells that are cancerous. And as you can see, basically any organ that you have could possibly undergo an autoimmune disease. The bummer with autoimmune diseases is they're not really well understood, which makes them really difficult to manage. So let's watch a video together. So let's take a walk through autoimmunity. This is going to talk about some of the factors that are known to contribute to autoimmunity, but as I said, this is an area where we just still don't know a whole lot of what causes autoimmune deficiency disorders and then how to manage them on the other side.
Through autoimmunity, the human body is a haven for life. And much like a prosperous country, is prone to attack. Throughout your entire life, every minute of every day, your body is under invasion from the outside world. Bacteria, microbes, toxins, and parasites have their own specialized methods of attack. Thankfully, your body can defend itself with its own highly sophisticated army, the immune system. Like an army, the immune system is highly diversified, full of specialized warriors capable of countering an attack, however and wherever invasion occurs. On the front lines of the battle, your skin provides a stalwart barrier between outside invasion and the fragile, life-sustaining organs within. To fight...
We call that part of your innate immune system. I know this thing's a little hokey, but it's good, it's good. And hopefully you recognize some of those warriors now.
Off attackers, also known as antigens, that do enter the body. The immune system wields an immense arsenal of cells which work together to prevent disease. Dendritic cells capture antigens and present them to T cells. T cells are the most important immune cells. They can recognize disease-causing microorganisms and rally other immune cells to attack the invaders or kill the invaders themselves. B cells produce antibodies, substances that are designed to attack specific antigens. Phagocytes, like ravenous monsters working for the immune system, are large cells that eat antigens, capturing and destroying microbes and other foreign particles. A healthy healthy immune system will keep the body free of sickness most of the time, recognizing harmful foreign invasion and using its legions of cells to attack sickness-causing antigens. But sometimes, the immune system stops working properly, and instead of attacking harmful intruding antigens, it mistakenly attacks the body's own cells. This friendly fire is called autoimmunity. In an autoimmune disorder, the immune system begins to manufacture T cells and antibodies directed against its own cells and tissues. As a result, healthy cells and tissues are destroyed, which leaves the person's body unable to perform important functions. Nobody is certain exactly why or how autoimmune disorders occur, but contributing factors include genetics and environmental conditions that can trigger the disorder. Some of the known environmental triggers include drugs, viruses, bacteria, foods, pollutants, hormones, and stress. There are hundreds of known autoimmune disorders that affect nearly every part of the body, but some of the most common are rheumatoid arthritis, lupus, Crohn's disease, psoriasis, multiple sclerosis, and vasculitis. Treatments for autoimmune disease include hormone replacement. In some autoimmune diseases, hormone-producing glands are destroyed, leaving the body without key parts of its messaging system. When this occurs, a doctor can prescribe replacement hormones. A common example of this is in the use of insulin in patients with type 1 diabetes or thyroid hormone replacement in Hashimoto's disease. Corticosteroids are used to control inflammation by suppressing the overactive immune system. The disadvantage of this treatment is that it can leave the body susceptible to outside harm because they suppress the whole immune system. NSAIDs, non-steroidal anti-inflammatory drugs, are used to ease pain and swelling. DMARDs, disease-modifying anti-rheumatic drugs, are used to suppress an overactive immune system and can reduce pain and swelling and prevent long-term damage to joints and organs. Biologics, often called immune response modifiers, are a class of drug which interpret the events that drive inflammation by targeting a specific attack rather than a whole body attack. Currently, scientists are experimenting with stem cell therapy treatments for autoimmune diseases. Such treatments involve reprogramming cells to replace damaged tissue, or replace a patient's immune system altogether by replacing bone marrow, where immune cells are generated. Though millions suffer from autoimmune disorders, more new treatments and research are available to keep the body's defenses aimed towards its enemies rather than towards itself. Brought to you by the American Autoimmune Related.
You might recognize at the center of this autoimmune disease graphic, that guy, right? Virus. So viruses are one of the things they think might trigger or cause an autoimmune disease, in that sometimes an antigen from a virus can look so similar to, can look really similar to a self-antigen. And so now cells have been trained for this viral antigen, but that viral antigen looks really close to a self-antigen. And so now those cells are attacking self instead of attacking self. In addition to that virus, they're also attacking self cells. So that's one of the thoughts behind what is happening with autoimmune diseases. And there certainly does seem to be a genetic component that if, for example, rheumatoid arthritis, if you have a grandparent that has it or a parent that has it, you are more likely to have it. I mentioned this earlier, but I thought this would be a good time to talk about positive versus negative selection of those T cells. So we talked about how before those cells get into the lymph system for their training from a naive cell into an active effector cell, like a naive CD8 cell would become a mature cytotoxic cell. While they're still in, before they get to the lymph system, they would undergo positive and negative selection. So here's part one. Positive selection. T cells must recognize self-major histocompatibility proteins, or those MHC proteins. So here's what you're looking at. Here's the self cell and the other side of that, and that MHC protein. And here's our T cell and the T cell receptor. So this T cell receptor has to be able to recognize this MHC. So in this case, not a good match, right? Shapes don't fit. Here, puzzle pieces match. We have the MHC and the receptor on the T cell matching. If they fail to recognize, then this undergoes cell death, right? It's not going to work if you can't, a T cell is not going to work if it doesn't recognize the MHC complex. If it recognizes the MHC complex, that's great. You get to go to the next level up of training, and that is called negative selection. So this is positive selection because it has to recognize it. There has to be a positive connection there. Here's the opposite. T cells must not recognize self-antigens. If the T cell recognizes self-antigens, then it's going to poke holes in it and kill it. That is not what we want our T cells to do. That is an autoimmune disease. So in this case, you can see there's no little gap here. So this orange oval is the self-antigen, and in this case it's all beautiful perfect match. Recognizing that self-antigen leads to apoptosis of the T cell. It's going to die, which is good because if it didn't die, then it could cause an autoimmune disease. And this is one of the cases they think, well, maybe some of these things are getting through that screening process, and that could cause an autoimmune disease. It should fail this test. It should not make a good connection here with this antigen. And if it doesn't make a good connection with that antigen, then this cell gets to continue on to its next level of training when it's going to hang out with that dendritic cell inside of the lymph node to become activated. Here it is even bigger. Positive selection, negative selection. Okay. Another way our immune cell systems can be a problem is allergies. So allergies are when our immune systems are attacking antigens that don't cause disease. So autoimmune, we're attacking self cells. Allergies, antigens that don't cause disease. Let's check this out. And going to. And for some of us, we are entering allergy season. One of the major, right? You can get an allergy from a lot of different molecules that are around in our environment, but pollen is certainly one of them that causes lots of people issues. So let's watch this brief video.
Allergens like pollen are nothing more than foreign plant antigens. When they get released into the air, you can see and hear the result. When allergens first encounter nasal tissue, sneezing is triggered. This is part of the body's immune defense. Pollen allergens then encounter the plasma cells in the nose, which respond by producing antibodies. These antibodies attach to mast cells, which are white blood cells containing the chemical histamine. As more antibodies are produced, they cause the mast cells to release histamine, which produces allergy symptoms such as stuffy and runny nose, sneezing, and watery eyes. These help to remove the invading pollen. Medications called antihistamines can be used to help relieve severe allergy symptoms.
It's a great sneeze. So if allergies get bad, progress, they can develop into asthma. Is one way people can get asthma is through allergies. And wow, asthma and your immune system. Let's check this out together. I think just seeing a bit of the complexity of that interaction between our immune systems and how asthma happens is pretty fascinating. And you'll recognize some of these players now that we've been learning about them as a class.
The lungs are where the body draws in vital oxygen and exchanges it for carbon dioxide. But every breath we take is accompanied by potentially harmful microbes, as well as particulates which can damage delicate lung tissue. To counter these threats, the lungs are enriched in immune cells that repulse invaders and repair injury. But this potent arsenal comes at a heavy cost. Overreactions or inappropriate responses result in diseases such as asthma. The lungs are spongy organs composed of increasingly finer tubules. First comes the trachea and the bronchi. These are then divided into the bronchioles, which are wrapped in a muscular sheath. At the ends of the bronchioles are microscopic air-filled sacs called alveoli. In the case of asthma, most of the harmful immunological events occur within the bronchi and bronchioles. The bronchial wall is made up of an epithelium covered in tiny hair-like structures called cilia, an interstitium, and muscle cells. Goblet cells scattered along the epithelium discharge a thin layer of mucus into the bronchiole lumen. The mucus is continually swept back up through the lungs by the action of the cilia. This system is known as the mucus escalator and is important for removing inhaled particles trapped within the mucus. Embedded throughout the bronchiole wall are immune cells such as dendritic cells, which sample antigens from the lumen, mast cells, which are packed with granules, tiny packages rich in histamine, and innate lymphoid cells, or ILCs. Individuals with asthma need to have been previously sensitized to a specific environmental antigen. The most common is found in the feces of house dust mites. But symptoms can also be worsened by respiratory virus infections, smoke, or pollutants. When inhaled antigens contact the epithelium, they trigger the release of two chemicals, IL-25 and TSLP, which stimulate nearby dendritic cells and innate lymphoid cells, those ILCs. The dendritic cells leave the epithelium for the lymph nodes, where they activate T cells and initiate an adaptive immune response. Antigen can also pass through the epithelium, especially in conjunction with damaging particles such as those found within tobacco smoke or diesel fumes. Some of this antigen can reach the mast cells housed in smooth muscle tissue. The mast cells of asthmatic individuals are unusual. They have more of an immune molecule called IgE stuck to their surface. The attached IgE then binds the relevant antigen, in this case from house dust mites, in a highly specific manner. When an antigen gets bound to two IgE molecules, it signals the mast cell to degranulate and spill out the contents of the granules. The granules' innards then exert their effects on surrounding cells and tissues. In asthma, the granule contents cause smooth muscle contraction and mucosal edema, leading to narrowing of the bronchioles. Many anti-allergy drugs target this key process of degranulation. Back to those T cells in the lymph.
So those granules would include histamines. Some air traffic happening here.
Nodes. Once activated, T cells enter the interstitium and team up with ILCs to escalate the asthmatic response. They secrete.
And when she's talking about things like ILCs or ILs, whatever, those are all cytokines.
Chemical signals that recruit other immune cells, most critical of these being eosinophils. These cells play central roles in almost all allergic diseases. Eosinophils squeeze through the epithelium into the lumen and begin releasing their own inflammatory signals, including.
So you might remember from our blood lab that eosinophils are one of the white blood cells. We don't have very many of them, and their primary job is to get rid of parasitic worms, something we don't encounter too much in the United States, but they also play a role in allergies. And a couple of you asked actually questions about eosinophils and allergies. And there's below this module, there's some connections, more background information for things for you to explore. And one of those links talks about this, if it's something you're more interested in learning about.
Including IL-5, IL-13, and platelet activating factor. Together, these act on goblet cells, causing them to ramp up production of mucus in a process known as goblet hyperplasia. By now, the victim is in the midst of an asthma attack. As the attack proceeds, more and more eosinophils are recruited into the lumen, and the mucus layer progressively thickens. The collective effects of smooth muscle contraction and mucus overproduction restrict airflow and result in the classic symptoms of asthma, including difficulty breathing, coughing, and wheezing. Multiple attacks can, over time, lead to fibrosis and permanent injury of the lung.
Pretty incredible, right? Just all the pieces and all of the cells that are involved in that asthma attack that lead to bronchial contraction, right? Those histamines are causing the contraction of that smooth muscle. And then those eosinophils that get activated are then causing more mucus to build up. Right, you saw other players in there like mast cells. All right, anaphylaxis. Some of you asked, what's the difference between anaphylaxis and allergies? So this would be a kind of a special case of allergies, especially intense case of allergies. And got another video to watch just because they have these some great animations and explanations. This one, I will admit, is, well, I'll let you make your own judgments. I think it's informative. We'll learn some stuff.
There's this idea that in portraiture...
Hold on.
Anaphylaxis. Anaphylaxis is a severe life-threatening allergic reaction which could result from exposure to any of the following causative agents: food, an insect bite or sting, drugs, latex rubber, exercise, idiopathic. On exposure, allergen molecules or antigens enter the bloodstream, where they interact with sensitized mast cells, to which IgE antibodies are bound. The allergen binds to the IgE, activating the mast cell, resulting in its degranulation. Mast cells degranulate, resulting in the release of the following inflammatory mediators: histamine, prostaglandins, leukotrienes, chemokines, platelet activating factor, and heparin. Histamine in particular binds to the H1 receptor sites in the endothelium, activating them, resulting in the following: itching and hives due to the increased vasodilatation, angioedema due to the increased vascular permeability, bronchoconstriction due to the contraction of the smooth muscles of the airway. bradycardia, and hypotension due to the increased vasodilatation. Epinephrine injected into the muscle is the first line in the treatment of anaphylaxis. It acts by binding to the adrenergic receptors in the endothelium, thereby countering the effects of histamine and the other inflammatory mediators. A second dose of epinephrine may be required if there is no improvement in symptoms within 5 minutes of administration of the first dose. Oral antihistamines may be used as adjunct therapy in the treatment of itching and hives. They act by competitively binding to the H1 receptor, thus preventing the circulating histamine from doing the same and exerting their effect.
So anaphylaxis tends to happen when the antigen that is causing the reaction, this extreme allergic reaction, is in the blood system, and then you have this massive allergic reaction that causes anaphylaxis, which can be deadly since it swells up the tongue so you can't breathe and makes it difficult to breathe due to the bronchial constriction. So ways to improve your immune system. Laureen talked about this in her talk as well, in our conversation about COVID. I liked this little cartoon reminders of different things you can do to help your immune system, which are similar to just helping your overall health. So I'll let you review that on your own. But let's talk specifically about what sleep deprivation does to your immune system in fighting a virus. So this is a MedCram video. You've also got a link to the MedCram video that talks about the acute respiratory distress syndrome and how that happens with COVID-19. But this one does a really nice job talking about what is it about sleep deprivation that causes our immune systems to not work so well. I think it's pretty fascinating. If you're liking this kind of stuff, there are several MedCram updates on COVID-19 if you're looking for ways to fill your time. But let's watch this one.
And we are going to move on to other things, but I wanted to restate and re-underline the importance of rest and sleep in terms of the activation of the immune system, especially at a time like now where people are very concerned. They may not be getting enough sleep. It's important for us to not underestimate the benefits of sleep. Now, we're just going to review a little bit, but if you want to know more in detail, go back to update 17 on the coronavirus that we've done. But real briefly, this was a study that looked at the effect of sleep deprivation on response to immunization. The participants were sleep deprived and given a vaccine to the influenza virus. And those that were not sleep deprived had a much better immune response in terms of antibodies to the vaccine. And the conclusions that the authors drew were, these results indicate that the response to the influenza vaccination is likely to be impaired in individuals who suffer from chronic partial sleep restriction. Because adults who show poor responses to vaccines and other antigenic challenges also experience higher rates of clinical illness, our findings support the concept that adequate amounts of sleep are needed to maintain resistance to an infectious challenge, such as COVID-19. This is the other study that we looked at in Update 17, and it looked at the sleep habits, how long they slept, and what was the efficiency of their sleep in terms of 14-day baseline. And then they went ahead and dropped rhinovirus-filled drops into the nasopharynx of these subjects, and they waited to see how many of them would develop the common cold symptoms. And what they found was quite astounding. They found that those with an average sleep duration of less than 7 hours were not 10% more or 50% more or even 100% more likely to develop the cold, but actually almost 3 times more likely to develop a cold. And when they looked for confounders, these things could not be explained in terms of differences in pre-challenge virus-specific antibody, demographics, season of the year, body mass, socioeconomic status, psychological variables, or health practices. And so here we have, at least for rhinovirus, evidence that poor sleep efficiency and shorter sleep duration in the weeks preceding an exposure were associated with a lower resistance to the viral illness. But there's even more research that just came out last year, and this is the focus of sleep that I want to look at today. So the first thing that you have to understand is that there is a cell which is infected with a virus, and in this case, it was a CMV infected cell. CMV stands for cytomegalovirus. It's a common virus that can cause a chronic infection. Here we have the nucleus. And because this cell is infected with cytomegalovirus, it makes its normal proteins on the cell surface so that the cells of the immune system would recognize it. But it also puts a little portion of that cytomegalovirus protein out on the surface to let the immune cells know that it's a infected cell. And so these regular proteins that are made in the cell are known as an MHC1. That's not that important to understand, but that's the name of the protein. This here is a portion of the virus that lets the immune system know that it is in fact an infected cell. There are other proteins that are also on the cell, and these proteins help immune cells dock with them so that they can make the appropriate determination of whether or not this cell should be destroyed or should survive. And this protein is known as ICAM. I-C-A-M. So far, so good. Now, let's bring in our immune cell. It's our cytotoxic T cell. It's responsible for going around and making sure that cells are not infected and taking care of cells that are infected. And these would be the cells that would be going around and getting rid of coronavirus infected cells so they don't produce more virus to infect more cells. And in fact, if this can be done early in the course, you would have a limited disease, and it wouldn't lead to many, many cells being infected, causing many T cells to have to try to fight and causing that cytokine storm. Well, there's a couple of proteins on these T cells. So this is a T cell, and it's a cytotoxic T cell. Not toxic to you, but cytotoxic to the cell that's infected. These are actually your friends. So there is a receptor on the T cell that sees that little particle of the virus and understands that this cell needs to be destroyed, and that is called the T cell receptor, or the TCR. The TCR also activates right next to it, and I'll show this here in green, that it's activating the beta integrin. This interaction between beta integrins and the ICAMs allow for a very strong docking that allows these cells to bind very, very strongly to these infected cells. And that allows these T cells to secrete cytokines that will shut down and kill the cell before it produces more viruses. So it's very important that the beta integrins and the ICAMs are able to bind together. Normally, the beta integrins are not activated, and they will not bind these ICAMs. Otherwise, your immune system would start to destroy your own body. So they're shut down. But there are certain situations where they need to be turned on. And if they are not turned on, your immune system is not going to work very well. Well, in this case, it is the TCR which activates these beta integrins and allows this to occur. The purpose of this article was to see whether or not things can affect this turning on of the beta integrins. And sure enough, they discovered that there is a receptor called the G-alpha S receptor. And this receptor is coupled to what is known as a G protein, and it's coupled to an enzyme known as adenylate cyclase. There's a lot of moving parts to this receptor, but I'm going to simplify it and show you that essentially it takes ATP, which is abundant in the cell, and it converts it into something called cyclic AMP. And as it turns out, cyclic AMP shuts down the ability of the TCR to activate the beta integrase. So the question is what is actually stimulating this G alpha S receptor? There's a number of things that can activate this. One of them is epinephrine, typically dopamine, histamine, serotonin. And the thing about all of these things that can actually stimulate this receptor here, they're all quite elevated when you are awake. And so you can see it's possible that epinephrine, dopamine, histamine, serotonin could stimulate this receptor, which would eventually convert ATP into cyclic AMP, which would then inhibit TCR's ability to have the beta integrin bind with ICAM and to have this T cell destroy with cytokines this infected cell. So is it possible that sleep could stop epinephrine, dopamine, histamine, and serotonin from binding this receptor and allow the beta integrin and the ICAM to bind tightly so that the immune response would be more robust? And so now the title becomes a little bit more understandable. GAS, coupled receptor signaling, and sleep regulate integrin activation of human antigen-specific T cells. And this was published last year in the Journal of Experimental Medicine. They infected these with the Epstein-Barr virus as well. And so we've got data for both of those. So here we can see the data for CMV-specific CD8 cytotoxic T cells. They looked at isoproterenol, which does hit that receptor, epinephrine, which does hit the receptor, and norepinephrine as well. And here on the x-axis, as we increase the concentration, you can see that that effect is that the binding of ICAM1 as a percentage of the control starts to drop off precipitously. And you can see that here with prostaglandins as well as adenosine. In the EBV, or the Epstein-Barr virus cell cultures that were infected, as the increased concentration of the same substances that we talked about, you can see that the ICAMs do drop. So it's one thing to say that these specific substances make it drop, but the question is whether or not sleep is actually beneficial in making sure that the cells can bind to their targets and take them out. And that's what they attempted to show here in human patients. What we see here, again, is the same thing on the y-axis. We're looking at ICAM binding. But we have two types of patients in this scenario. We have the ones that are filled in with black circles, and these were the ones that were sleeping at night and awake during the day, as opposed to these open circles, which are white in the middle, where they're awake during the night and also awake during the day. And so you can see the effects here in the CMV infected cells on the left and the EBV infected cells on the right. In the nighttime, which is shaded here and over here as well, at least at 2 o'clock in the morning and here at 6 o'clock in the morning, there was a statistically significant increase in ICAM binding in these patients that were sleeping at 2 o'clock in the morning as opposed to being awake at 2 o'clock in the morning. And I just wanted to point out for those of you who are doing shift work that so long as you are working at night and going home and sleeping with your lights off and no light coming in through the windows, and you do this every single day throughout the weekend, you can also get the same benefit from getting sleep even though it's when the sun is up. And then they decided to look at that over the different types of differentiation of these cytotoxic T cells. They looked at ones that were early in their life, intermediate in the cell life, and late in the cell life. And what they found was this effect seemed to be the strongest in the early phase of the cell's life, despite the fact that it didn't have a lot of these G receptors that we talked about. That might suggest that it's the early cells that are being amplified against the infection at hand that is more influenced by the effect of sleep versus wake.
So just to make sure that we're all following this together, because graphs, the higher this line is, the better the cytotoxic T cell is binding to the infected cell, and they're binding closer together, which means that the cytotoxic T cell can effectively get this infected cell to die. Greater binding means better cell death, and you can fight the virus better. So when we're sleeping, our T cells, our cytotoxic T cells, are better at binding to cells that have been infected with a virus, and can kill them easier. Why? Because when we're asleep, our sympathetic nervous system calms down. There's less epinephrine around. There's less of those other binding agents to those cell receptors, and we're just able to bind those better. Isn't that incredible? Man. And they just figured this out last year, the mechanics of it.
Of sleep versus wake. So the conclusions of the authors are this. We show here that several G-alpha-S coupled receptor ligands inhibit TCR-mediated integrin activation on antigen-specific CD8 T cells. These are the ones that are doing the killing here. Whereas sleep upregulates integrin activation by suppressing G-alpha-S coupled receptor signaling. Given the importance of integrin activation for the formation of immunological synapses, our data suggest a critical role of conditions like sleep that are characterized by low-level G-alpha-S coupled receptor ligands in boosting the T cell responses. And I'll tell you, it's those T cell responses that are very important in your immune system getting rid of coronavirus infected cells. So you can imagine the current scenario, and it doesn't take a lot of drawing on here to modify this picture for the current COVID-19 situation. Sleep is important, and you don't have to go to the store to buy it, and you don't have to wait for a pharmacist to dispense it. It's something that we can do. Put down your iPads, put down your phones, and go to sleep, and wake up with a better immune system. For those of you who have difficulty sleeping, if you have insomnia, we have a free course at medcram.com on how to deal with insomnia. So please check that out. Thanks for joining us.
Well, thanks for having us. There's a really simple drawing. You get enough sleep. These G-alpha coupled receptors, there's less, what are they calling? Plasma mediators like adrenaline. Fewer of those around, that doesn't get activated. If that doesn't get activated, then you get better connection here between our cytotoxic T cell and the cell that is infected, and your immune system can fight that off. Sleep deprivation, lots of adrenaline still around, lots of binding to this G-alpha receptor. That G-alpha receptor, when it's bound, sets off that secondary messaging system, which basically doesn't allow that binding between our cytotoxic T cell and the infected cell. And the infected cell continues to survive and produce more viruses that can then go infect more cells. So you can check out the MedCram course, the free course. I haven't checked it out. There's all kinds of great stuff online. One of them is the sleepfoundation.org, and they have a whole page on sleep hygiene. So tips and tools to help you if you are currently not getting your 7 to 8 hours of sleep every night, some things you can do to help your body do that. So I encourage you, if you're not getting the sleep you need, figure out how you can. It's one of the most important things you can do for your life, not just for fighting off COVID-19, but sleep's also one of those things that helps get rid of chronic inflammation. So it's good to have great sleep habits. So let's do some review questions. HIV targets and kills helper T cells. What effect does this have on cytotoxic T cells? So this is a multiple choice question, but one of the great strategies you have, and we've talked about this semester, is you read the question first, figure out what you think the answer might be, and then read through the actual, your options for multiple choice. So if we don't have helper T cells, what's that going to do for our cytotoxic T cells? Do you remember what that role was for helper T cells? What were they helping our cytotoxic T cells to do? All right, let's look at our options. A, cytotoxic cells would not be able to bind to their target cells and kill them. So do you need the helper T cells there when that binding between the cytotoxic cell and the infected cell happens? B, cytotoxic cells could not release their toxic compounds to infest, to kill infested cells. So those granular compounds. C, cytotoxic cells could not be activated. And remember, where do they get activated? Yes, in the lymph nodes. And D, cytotoxic cells could not make their MHC1 proteins. So what do you think? What did you say here? And did you find it here? Because that's the game. Ooh, wrong game. If you said C, as in Cocoa Puffs, cranium, crayons, you're correct. You need helper T cells in order for your cytotoxic cells to be activated, start dividing, and become mature cells. Cytotoxic T cells recognize their targets, sorry about the grammar there, using what? MHC2 proteins, MHC1 proteins, cytokines, or unicorns. B is in beautiful, MHC1 proteins. Sleep deprivation weakens the immune system to fight a virus by, I'll let you read through those. Well, I hydrate. Also important for your immune system. Are you ready? All right, if you need more time, hit the pause. Because the answer is, let me remember, B. Epinephrine binds to those G-alpha receptors, which reduces the ability of our cytotoxic T cells to bind to their target cells and kill them. So get some sleep. Here are some pictures to inspire you. Great to have you in class. If you have questions, please let me know. Today, for me, as I am recording this is Monday, April 13th. My next step is to write and launch your immune system assessment, and that will be due the 24th of April. Sleep well, my friends. Fight off that virus.